The process of cell division. Learning objectives. Explain the role of chromosomes during cell division. Describe the main events of the cell cycle. Describe what happens during the phases of mitosis. And investigate how daughter cells split apart after mitosis. Cell division takes place all the time, keeping us healthy by replacing worn cells and regenerating the tissues we lost to injury or disease. What would happen if the cell were simply to split into two, without any advanced preparation? The results might be disastrous, especially if some of the cell's essential genetic information wound up in one of the daughter cells but not in the other. In order to make sure this doesn't happen, cells first must make a complete copy of their genetic information before cell division can begin. Genetic information is bundled into packages of DNA known as chromosomes. Prokaryotic cells lack membrane-bound nuclei and many of the organelles found in eukaryotes. The DNA molecules of prokaryote cells are found in the cytoplasm, along with most of the other contents of the cell. Most prokaryotes contain a single circular DNA chromosome. That circular DNA contains all or nearly all of the cell's genetic information. Eukaryotic cells generally have much more DNA than prokaryote cells have, and therefore they contain multiple chromosomes. For example, fruit flies have 8 chromosomes per cell, carrot cells have 18, and human cells have 46 per cell. The chromosomes in eukaryotic cells contain DNA tightly bound by proteins called histones. This complexity of DNA and protein is referred to as chromatin. Chromosomes are precisely separated into 2 daughter cells during cell division. The prokaryotic cell cycle is a regular pattern of growth, DNA replication, and cell division that can take place very rapidly under ideal conditions. The process of cell division in prokaryotes is a form of asexual reproduction known as binary fission. Once the chromosome has been replicated, the 2 DNA molecules attach to different regions of the cell membrane. A network of fibers forms between them, stretching from one side of the cell to the other. These fibers constrict and the cell is pinched inward, dividing the cytoplasm and chromosomes between 2 newly formed cells. Binary fission results in the production of 2 genetically identical cells. Eukaryotic cells have a much more complex cell cycle than prokaryotic cells. During the cell cycle, a cell grows, prepares for division, and then divides to form 2 new daughter cells. The first part of the cell cycle is called interphase. This is known as the in-between phase and is divided into 3 phases, G1, S, and G2. G1 is cell growth. Cells do most of their growing during the G1 phase. In this phase, cells increase in size and synthesize new proteins and organelles. The G in G1 and G2 stands for gap, but the G1 and G2 phases are actually periods of intense growth and activity. S is the next phase and is for DNA replication. The G1 phase is followed by the S phase. The S stands for synthesis. During the S phase, the DNA is synthesized as the chromosomes are replicated. By the end of the S phase, the cell contains twice as much DNA as it did in the beginning of the phase. G2 is preparing for cell division. When DNA replication is completed, the cell enters the G2 phase. G2 is usually the shortest of the 3 phases. During the G2 phase, many of the organelles and molecules required cell division are reproduced. When the events of the G2 phase are completed, the cell is ready to enter the M phase and begin the process of cell division. The M phase of the cell cycle, which follows interphase, produces two daughter cells. The M phase takes its name from the process of mitosis. During the normal cell cycle, interphase can be quite long. In contrast, the process of cell division usually takes place very quickly. In eukaryote cells, cell division occurs in two main stages. The first stage of the process, the division of the cell nucleus, is called mitosis. The second stage, the division of the cytoplasm, is called cytokinesis. In many cells, the two stages may overlap, so that cytokinesis begins while mitosis is still taking place. Biologists divide the events of mitosis into four phases: prophase, metaphase, anaphase, and telophase. Depending on the type of cell, mitosis may last anywhere from a few minutes to several days. The first phase of mitosis is prophase. During prophase, the genetic material inside the nucleus condenses and duplicates. Chromosomes become visible. Each duplicated chromosome condenses to appear as two thick strands known as sister chromatids. They attach at a point called a centromere. When the process of mitosis is complete, the sister chromatids will have separated, one to each of the daughter cells. Also during prophase, the cell starts to build a spindle, a fan-like system of microtubules that will help to separate the duplicated chromosomes. Spindle fibers extend from a region called a centrosome, where tiny paired structures called centrioles are located. Early in prophase, the centrioles move toward opposite ends or poles of the cell. Plant cells lack centrioles and organize spindles directly from their centrosome regions. The second phase of mitosis is metaphase, and this is generally the shortest phase. During metaphase, the centromeres of the duplicated chromosomes line up across the center of the cell. Spindle fibers connect the centromere of each chromosome to the two poles of the spindle. The cell is now ready to separate those sister chromatids. The third phase of mitosis is anaphase. Anaphase begins when sister chromatids suddenly separate and begin to move apart. Once anaphase begins, each sister chromatid is now considered an individual chromosome. During anaphase, the chromosomes separate and move along spindle fibers to opposite ends of the cell. Anaphase comes to an end when this movement stops and the chromosomes are completely separated and into two groups. Following anaphase is telophase, the final phase of mitosis. During telophase, the chromosomes, which were distinct and condensed, begin to spread out into tangles of chromatid. A nuclear envelope reforms around each cluster of chromosomes, and gradually a nucleolus becomes visible in each daughter nucleus. Mitosis is complete, but the process of cell division has one more step. As a result of mitosis, two nuclei, each with a duplicate set of chromosomes, are formed. All that remains to complete the M phase of the cycle is cytokinesis, the division of the cytoplasm to form two separate cells. Cytokinesis usually occurs at the same time as telophase. Cytokinesis completes the process of cell division by dividing one cell into two, also known as two daughter cells. The process of cytokinesis differs in animal and plant cells. In animal cells, the cell membrane is drawn inward until the cytoplasm is pinched into two nearly equal parts. Each part contains its own nuclei and cytoplasmic organelles, as shown in the picture on the screen. Cytokinesis in plant cells proceeds differently. The cell membrane is not flexible enough to draw inward because of a rigid cell wall that surrounds it. Instead, a structure known as a cell plate forms halfway between the divided nuclei. The cell plate gradually develops into cell membranes that separate the two daughter cells. A cell wall then forms in between the two new membranes, completing the process. There's a picture of this on page 504 of your textbook. Mitosis overview. You should be able to list and describe the phases of mitosis. The phases of mitosis shown on this slide are typical of animal cells. For interphase, you should be able to identify that the cell grows and replicates its DNA and centrioles. Once it's finished interphase, it enters the M phase, or mitosis. The first phase of mitosis is prophase. For prophase, you should be able to identify that the chromatin condenses into chromosomes, the centrioles separate, and the spindle fibers begin to form. The nuclear envelope breaks down. The second phase of mitosis is metaphase. For metaphase, you should be able to identify that chromosomes line up across the center of the cell. Each chromosome is connected to spindle fibers at the centromere. The third phase of mitosis is anaphase. For anaphase, you should be able to identify that the sister chromatids separate into individual chromosomes and are moving to opposite ends of the cell. The last phase of mitosis is telophase. For telophase, you should be able to identify that the chromosomes gather at opposite ends of the cells and lose their shapes, and two new nuclear envelopes form. To finish off mitosis, the last step is cytokinesis. For cytokinesis, you need to be able to identify that the cytoplasm pinches in half. Each daughter cell has an identical set of duplicate chromosomes.